right? Conventional ag is based on chemistry set experiments. They don't take into consideration that what these soil life do is they raise and lower pH all day long to solubilize nutrients.
In different parts of the root zone, even the same plant during the same day.
Right.
Totally different.
Same spot, in this same spot all day long. You can go from 5 to 7 to 5, up and down, in the same spot based on the acids and bases that the soil life is exuding and to raise and lower the pH to solubilize nutrients. So this biological system is a much more sophisticated feedback loop than the conventional ag system, which is basically a soluble nutrients in solution chemistry set. So we think pH is important because we want the most nutrients to be soluble, and the most nutrients are soluble in this 6.4 to 6.8 range. I went fast through that. I'm thinking a few people didn't quite.
6.4 to 6.8.
Is what is generally considered to be the appropriate pH for most crops. But that whole idea is predicated on the thought that there's no life in the soil. And when you understand, I'll just get a little pen here and draw it.
We had some soils that feel like a pH of 4, and we got pretty good crops.
Well, so what's going on? Your pH here
We had microbes.
could be 4.
Yeah.
Your pH here could be 5.
You're taking samples.
But then it'll be 7,
Yep.
and then it'll be 5 again, and this whole thing will happen in 24 hours. What happens in the rhizosphere, in the area where the life is flourishing, is the pH goes up and down all day long because the plant is feeding the soil life, so the soil life can solubilize the nutrients so they can feed the plant. This is the biological system. It's something like 3% of the species of soil life have so far been named. That leaves only 97% we don't even have names for yet. Do you know why we haven't named them yet?
We're focused our energy on
No, by our
You can't culture them in a lab.
Yeah.
They won't exist outside of nature. You can't take agar in a petri dish and culture these things. That's not where they live.
You can see it when we put the soil under a microscope.
You can see when you
different
put a skull on a
ones.
microscope.
Yep.
And you can do now some sophisticated things with DNA.
Yep.
But historically, microbiologists didn't even have names for most of what's underneath our feet because they couldn't study it because they couldn't culture it in a lab. Because it lives in nature, it doesn't live in a lab.
They cultured it, the soil, for years and thought it was sterile, so
Yeah.
that's why they concluded the chemical approach. But they were culturing at human body temperature with one food, putting a lid on the dish so there's no oxygen. They were culturing for pathogens, basically. So they were saying that these soils had no pathogens before they started.
Well, that's why most of what was found was pathogenic, and which why we considered why we put fungicide in our seed. Instead of putting colostrum on our seed, which gives our plants the ability to establish good, healthy gut flora, we put fungicide in their seed, which kills all their colostrum, which guarantees we're going to have colicky babies. How's that for you? Get colicky babies, they're going to need antibiotics, they're going to need fungicides, they're going to need insecticides because they're sickly. Right? That's the way to set them off wrong. Brilliant. Awesome way to take money from the farmers.
Yeah.
Train them to grow sick plants and then sell them all the products to make them sicker. And you suck all
They're
the...
doing the drugs for people.
What I was thinking about all of this business here
Yeah.
is monoculture over a long time will extract specific elements more than others,
Mhm.
and this will increase the likelihood of deficiencies in a profile.
Sure. And it'll decrease the spectrum of species living in the rhizosphere. The more different families of plants you've got, the more gut flora you've got, the broader the spectrum of species. And we'll talk tomorrow about some of the magic things that soil life can do, which violates, as far as I'm concerned, laws of chemistry and not physics. But what life can do is so exciting. It's so, so exciting. Yeah.
So what helpful information is a pH test offering us?
Very little. It basically is telling us how full the tank is. The more hydrogens you've got attached to the soil colloid, the more empty the tank is. And so the lower your pH is, 3.4 means you've got a really empty tank, which means you need to fill your tank. As far as I'm concerned, when I see all these red clay soils down here with no organic matter that are totally tight, I'm like, you guys could so do with a few minerals. You could so do with a few minerals. And if you got pastures and you can't afford it per acre, I understand that. But if you're growing vegetables, you could really, really benefit from a few minerals. I grew up on an organic farm. We didn't talk about minerals. No one told me about this stuff. We had pests and diseases year after year after year. We struggled to make a living. I filled the tank with minerals, and now my crop yields are dramatically higher. My pest pressure is down. My disease pressure is down. My flavor, my shelf life is up. My marketability, my bottom line economics, my time off, all improved. And this is what, east of the Mississippi where we get lots of rain, we've got weathered soils. And down here, you guys didn't get glaciation recently. So your soils are even more weathered. The last time you got new minerals distributed by nature was a long time ago. As I understand it, there's this ridge that goes across, about a third of the way up in Iowa, and I-40 passes through just next to that. And all the land north of there is worth 20 grand an acre, and all the land south of there is worth 12 grand an acre. And what that ridge is where the glacier stopped.
Yeah, the glaciers never came down here
That's my point.
at all.
And that's why you got a pH of 3.4, is because the soil, it's been raining, 40, 50 inches a year for the past how many 100,000 years?
Billion years.
What's that?
About a billion years.
Since the last glaciation occurred?
California and the Great Lakes have been up and down 19 times,
Yeah,
and we've been up once.
up once. So I don't want to sound like a northerner and make comments about southerners, but I'll just say your soils are really, really weathered. They're really, really, really weathered, and that means their tank is empty, which means they don't have a lot to put into the plant, right? Which means there's not a lot to put into your body, right? I mean, it's real simple, and we're going to talk about how to address this, right? We have to break for lunch at some point, but nature does this with rocks. Like I said, the Nile River does this with crushed up rocks from the glaciers crushing up the rocks. This is what the Ganges River does. Is it crushes up rocks and spreads them on the land. Right? That's all you need. Those are the basic ingredients you need to build up soil, are these basic elements that are in rocks. So I'm not saying you need to spend lots of money on fancy fertilizer. If we mimic nature, if we see how nature does it, we can actually systemically really improve things with really little bit of money, with a really small amount of money. This is what's going on in India. This is why the land is so weathered and weak. In Africa, we've got major places where there hasn't been glaciation recently. They don't have the alluvial plains, and volcanism doesn't. I mean, volcanism is your other option. Volcanoes blowing up, right? Mount St. Helens.
Yeah.
That's your other option for remineralization. And on a different kind of time scale, sure, nature will revitalize the planet. But if you're talking about a human time scale, if I'm raising kids and I've got worn out New England farmland, it's really important to me to put the minerals down now, because I want those minerals in my kids' bones when they're young. I don't want to wait 20 years for nature to figure it out slowly through bioaccumulator plants and things like that. Anyway, I didn't want to spend too much time on these topics, but I think they're important to cover. Yes,
I just want to try and say a couple things and then get you to revise them or.
go for it, please.
So, taking a pH test from a soil that's been abused or not used or neglected will give you a certain amount of information, but it might not actually tell you what minerals are present, because the biology might not be active enough to give you
To release them.
in plain available form. So that's one thing I'd like to use,
Yeah, comment.
proper commentary on. And then the other thing is, two of the ways in which you could alter the pH would be to simply put out the food for the microorganisms to feed the bacteria and the fungi to start stimulating things, and that would potentially change it
Yep.
as well. But also simply just putting, like if you were to install or introduce a perennial polyculture, then the plants would begin altering the pH themselves, right?
Gotta make sure I'm not using the wrong pen on the wrong board here.
There's three things there, I guess.
You got your clay. You got your bonding sites. Low pH means you get all hydrogens there. That means you don't have the calcium to release in the first place. So, sure, there is stuff locked up in here. Sure, if you take a mining assay and you dissolve the entire soil, right? So what the Albrecht style soil test does is tells you what's reasonably releasable through biological activity. It's not perfect. There's no perfect soil test, but it's not, the Albrecht soil test is not telling you what's available right now. It's what can life reasonably release, as I understand it. If you dissolve the whole soil entirely with a very strong acid, you will find boron in here. You will find cobalt in here. You will find calcium in here. But it's not reasonably easily released. So my critique of the soil food web model is that if you don't have a bunch of minerals relatively available in the first place, you don't have the food for the soil life to eat. So I didn't talk about
It's
minerals. Minerals, it's true.
I'm just
You
saying,
spent tens of thousands of dollars.
More soil than anyone in the U.S. over lots of thousands of acres and doing a lot of this work and putting out microbes like crazy. Minerals will limit the microbes because they're going to harvest them
M&Ms,
first, and it's only when
microbes
they
and
grow
minerals.
That they die that they plants are fed. So
Microbes are found, I mean, it's the life,
how you get the minerals?
it's a
is
living
one
system,
way,
so we
but-
understand the critical importance of life. However, minerals are foundational. I like to use the example of vitamin B12. Vitamin B12, people have heard about vitamin B12. If you're a vegetarian, so I've been told, sometimes your doctor will tell you that you need to supplement your diet with vitamin B12 because there's not enough in your plant-based diet. There's not enough vitamin B12 for you to function. B12 in science names is cyanocobalamin. Cyanocobalamin basically means one atom of cobalt with a few amino acids attached to it. So it's not the amino acids that are the limiting factor, it's the cobalt that's the limiting factor. So if you don't have enough vitamin B12, you become anemic. You become weak. You have low energy. You just, right? We know vegetarians,
Yes.
stereotypes,
No.
vegetarian, no
It
stereotypes.
happens.
It's not a stereotype,
It happens
it's
to a lot of
true.
people. Yeah.
Stereotypes are true. Not always, but a lot. When you really don't have enough vitamin B12, you know what happens? You become dead. Right? You don't need much vitamin B12, which is really cobalt. You don't need a lot. You need a really small amount. But if you don't have enough, you're dead. End of conversation. Exactly the same for microbes. Right? 80% of the 3.5 million species of microbes are B12 dependent. You don't have enough cobalt in the soil, they're dead. What I like to say is, if you were to take 2 airliners full of humans and drop them off in Alaska because you wanted to inoculate humans into the environment where there's no humans, they're mostly going to die. Right? They don't have the critical components necessary to live in that environment. That environment is inhospitable for them. So you can keep dropping them off. You can keep applying compost teas, and you apply more compost teas, and they're alive for a little while, and they start to build buildings, and then and the next time that you drop some more off, maybe they find the buildings that are halfway built, and they get them established, or the garden that got planted, but they died before the garden came to fruition. And so only adding inoculants without balancing the minerals as well, to me, is it's a silver bullet strategy. It's saying it's all microbes, which is no, microbes are really important, but you also need minerals, you also need air, you also need water, you also need organic matter. If you don't see the whole process as a living ecosystem and make sure that all the pieces are present, you're not looking at it honestly and dispassionately. So it is true that we have plants that are called bioaccumulator plants. And it is true, I mean, you can, the best way to get rid of dandelions is to add limestone, right? I mean, dandelions are a calcium bioaccumulator, and when you've got enough calcium in the soil, they'll go away. all these different plants we've got the docks, we've got the thistles, all these there'll be things we call weeds that people come in and try to kill with their herbicides. Those are nature's way of balancing minerals. So they are bioaccumulators of different elements, and they'll grow for a couple generations or 10 years until the minerals that they are bioaccumulators of are present in sufficient levels, and then they'll die off, and the next families of plants will come through. That's the way nature balances her soils, in between the ice ages, as I understand it. The question is, are they just pulling minerals out of the subsoil or something else going on? Because I'm aware of research where people did something like they took nettle, which is considered to be a boron bioaccumulator, and planted nettle in a pot of potting soil that didn't have any boron in it, and grew the nettle, and then tested the nettle after it grew and found the boron in the plant, even though it wasn't in the potting soil, and there was no subsoil for it to be bioaccumulating from. So, yes, there was boron in the nettle, and no, it did not come from the soil.
They did a grinding combustion on the soil.
I think so. Yeah. So Jerry Bernetti who told me this story. I don't know. I didn't track down all the details. This is a great one to test.
Because the boron's in every cell, so if there was soil, it's, like you said, it's in there, but it
It's
might not have been in the
Potting
in the
soil or
exchangeable,
whatever.
ready to go state.
There's all kinds of things you can do. I mean, it's leading towards this topic, which is, the formal term is biological transmutation, which you can test in many other ways as well. Anyway,
So isn't there,
on my agenda for tomorrow, but
So microbes with minerals work better, like we started adding.
M&Ms. And you know what M&Ms are? Microbes and minerals. Microbes and minerals.
enzymes.
Microbes and minerals.
So if you add
If you don't have
enzymes-
microbes and minerals, you're out of luck. As far as I'm concerned, you have to do both. You can't just do one. You can't just do minerals. You can't just do microbes. You need to understand where you're at in the balancing act.
so is that nettle story, defying the law of entropy?
I'm going to talk about it tomorrow. It's on my agenda for tomorrow. It just came up as part of this conversation in relation to bioaccumulator plants. To answer, my truth is there's a lot more going on there than we understand. And we have data that tells us there's a lot more going on there than we understand. Good, proper scientists have documented it over and over again in lots of different cases, and we don't like to talk about it because it doesn't fit with what we learned in science. But I was watching, we homeschool our kids, and we have a tutor comes, and she was talking to the kids about brains, and the brains on Thursday. That was her. She made a little brain cap, and then we had the different parts of the brain and what happens in this part, what happens in that part. And she was telling the kids about memories being in the brain, stored. And I know they're not stored in the brain. I've got 3 cousins that are PhD neuroscientists, and I ask my cousin Audrey when she comes to Thanksgiving every year, Audrey, have you guys found memories yet? And she has to say no, because they haven't.
They're stored in cells.
Well, where they're stored is a really interesting conversation. That's where I'm going with it, is the field. Because we think they're stored like information stored on a computer hard drive with ones and zeros, and they're not. They're not stored in your brain. Memories are not stored in your brain. That's what neuroscientists will tell you, even though you learn in school that they are. Right? So what's the cutting edge science has figured out won't get into school for 50 years. And so we're still trained about this stuff. So we are not trained about biological transmutation, even though it's good, solid literature that's decades old at this point. I really do need to break for lunch.
Yeah, now's a good time to refill our brains.
I need to break for lunch. I haven't gotten anywhere near as far as I want to on this topic,
Dan,
but
can I get you to clarify
I think
one
that's fine.
thing on-
I will clarify one thing and then I want to break for lunch.
All right.
Yes.
I heard you say 80% of soil microorganisms are dependent
Are
on B12.
B12
Do you mean they're
dependent.
dependent on cobalt to make B12, or they're dependent on B12 that they're
They need
sourcing from some other place?
B12 to function biochemically,
So they're-
and cobalt is
So they're
part-
dependent on cobalt
they're cobalt dependent
to
organisms.
make B12.
Yes,
okay.
you can't have B12 without cobalt.
Yeah.
Yes, they're cobalt dependent organisms. All right.